All right, good morning, everybody. It's my pleasure to introduce Samuel Wilson, who joined the Battle Act Hussey Laboratory as a Doris Duke undergrad this year. We've had 3 or 4 over the last couple of years, and Sam, you represented that whole scholarship wonderfully. I mean, when Sam came in, he immersed himself in a very challenging project involving the role of extracellular matrix in esophageal cancer, and he was involved in all aspects of what you're going to see today. In particular, pay attention to some of the microscopy photos. It's a world premiere of MBV uptake in cancer cells. It's quite amazing. So with no further ado, I give you Sam Wilson.
All right, hello, my name's Samuel. I was a summer research intern at the McGowan Institute for Regenerative Medicine through the UPMC Human Cancer Center and the Doris Duke Foundation. My research for the summer was with the Hussey Lab. We were focused on characterizing interactions between the MBV and esophageal epithelial cancer and normal cells. We were looking at esophageal adenocarcinoma. It's one of 2 subtypes of esophageal cancer, and it's one of the most common causes of cancer death in the world, with a 21% 5-year survival rate. You can see here in figure A, this is the normal squamous state of your epithelium in the mucosa. It's composed of stratified squamous epithelial cells, but they have high plasticity, so changes in the environment like acid reflux can cause it to become Barrett's esophagus, like in figure B. These cells are now a single columnar layer of cells with mucus-producing glands. These cells are even more highly plastic, so they can change even more and more and more in figure C, D, and E until they become eventually adenocarcinoma. And this cancer doesn't have too many effective treatments as of right now. So we're looking at matrix-bound nanovesicles, or MBV, as a possible therapeutic option. These MBV are attached to the extracellular matrix produced by different cell types, and they've already shown therapeutic and modulating effects in other disease states and cell types. They've shown it can activate bone marrow-derived macrophages towards an anti-inflammatory phenotype, stimulate cell proliferation of human perivascular stem cells, and reduce the effects of racine-induced rheumatoid arthritis. We want to see what the effects of these MBV are on esophageal epithelial cells using MBV derived from urinary bladder extracellular matrix, or UBM. Our hypothesis is that the UBM MBV can mitigate neoplastic esophageal cell phenotypes, meaning they can reverse the phenotypic changes of the epithelial cells within the esophagus from adenocarcinoma to a more normal state, which could include Barrett's esophagus or the normal squamous state. We had 2 objectives. We were first testing the in vitro effect of the UBM MBV in normal and cancer esophageal epithelial cells, and then we were characterizing those in vitro interactions. For our first objective, we used the HET1A and SKGT4 cell lines, 2 cell lines derived from human esophagus, one with and one without adenocarcinoma. We kept them in an 8-well microslide, as seen here, incubated overnight until they were 50% confluent, and then treated them with media control, or the UBM MBV, for 24 and 48 hours. For our outcomes, we were looking at confluency and viability. Confluency using bright field microscopy, and then viability using a live dead cell imaging assay. You can see here in our non-malignant and neoplastic cells, after 24 hours in the media control, the cells have proliferated, they've grown, and they've attached to the surface of the wells. This is after 24 hours of treatment. In the media control, and then after 24 hours of treatment, you can see changes in the morphology. Changes in the morphology, as well as reductions in confluency for both cell types, with a more dramatic reduction in the SKGT4 cells. After 48 hours, the cells proliferated more in the media control. You can see them attached to the cells and attached to the well and branching out towards one another. But after 48 hours of treatment, you can see a more dramatic reduction in confluency for the SKGT4 cells, as well as the cells starting to look detached from the bottom of the well, suggesting that these cells are most likely dead. We then ran a live dead cell imaging assay to confirm whether or not the viability of these cells was changing. In green, you have the living cells, and in red, you'll have the cells positive for cell death. You can see in the media control and in the UVM MBV treated groups of the HET1A non-malignant cells, there's no dramatic or obvious change in the number of viable cells. But in your SKGT4 cells, you can see a huge reduction in the number of viable cells per field of view. We then quantified this data, and we see that there's a significant decrease in the percentage of viable cells after 48 hours of treatment with the MBV, and no significant difference in the normal HET1A cells. This showed us that our initial hypothesis was incorrect. It's not that that the MBV are changing the phenotypes of these cells to a more normal state. It's instead killing. So then we were looking at characterizing the form of cell death caused by the UVM MBV. In this particular study, we were looking at apoptosis to see if that's what the MBV were causing. We used the same cell lines and an 8-well microslide, incubated them overnight, and then treated them with either media, the UVM MBV in media, and staurosporin in media. Staurosporin is a chemical that induces apoptosis in cells. For our outcomes, we were looking at apoptosis using a NuQ 488 reagent, which is a reagent that's bound to a dye that when it enters cells undergoing apoptosis, enzymes that are in charge of apoptosis will clip that dye, allowing it to go and bind to the DNA, showing us when cells are undergoing apoptosis. Here you can see in our negative control, which is just the cells treated with media and left for 24 hours. In your HET1A cells, you're still going to see cells undergoing apoptosis just from the natural cell cycle. And for your SKGT4 cells, these are neoplastic cells, cancer cells, meaning they're going to avoid apoptosis as much as possible to continue proliferating. You can see in our positive control, when we induce apoptosis, you get a different result with a lot of cells undergoing apoptosis in both SKGT4 and in the HET1A groups. But you see in our UVM-MBV treatment, there's no dramatic difference between the negative control of the HET1A cells and the UVM-MBV. But for the SKGT4 cells, you can see an increase in the number of cells per field of view undergoing apoptosis. This suggests to us that the UVM-MBV at least partially contributes to apoptosis in our neoplastic cells. Then we were looking at characterizing the form of cellular uptake the UVM-MBV are being used by the epithelial cells. We first isolated our urinary bladder extracellular matrix UVM-MBV using differential centrifugation, filtration, and ultracentrifugation just to remove the MBV from the ECM. We then labeled these with a surface protein staining dye called CFSE, and then ran them through a size exclusion chromatography column in order to remove excess dye from our solution. After that, we put them through two different studies, a dose response study and a time course study. In our dose response study, we treated them with either a PBS and CFSC labeled control or our CFSC labeled MBV at 3 different concentrations of 4.6 e to the 11th, 2.3 e to the 11th, or 1.15 e to the 11th particles per milliliter for 1 hour. In our time course study, we treated them with either the PBS control or the labeled MBV for 1 hour, 30 minutes, and 15 minutes. For outcomes in our dose response study, we were looking at uptake via fluorescent microscopy and quantification via ImageJ. In our time course study, we were just looking at uptake via fluorescent microscopy. In our dose response study, you can see in red the actin stain and the nuclei stained in blue, and the MBV would be in green with the CFSC labeled dye. In our control with just the CFSC and PBS, it's hard to see there, but there is green in the bottom over here. But that's just the dye itself being shown here. In our lowest concentration, 1.5 times e to the 11th, you can see a higher intensity here showing that the cells are positive for MBV. This is in our non-malignant HET1A cells. In our 50%, you can see that intensity is similar to the control. We're not sure why, but this was constant across both replications we did. It might have something to do with the cell cycle, whether or not the MBV can be uptaken by the different cells in the different stages they are in the cell cycle. We're not too sure. We're going to keep looking into that. But in our highest concentration, not only can you see the cells are positive for the MBV, you can also start to see a clear endocytosis vesicular response within the cells, vesicular pattern within the cells, showing the cells are undergoing endocytosis. For our SKGT4 cells, you're going to see the same thing for the control, where you just see the dye itself within the cell. In our lowest concentration, middle concentration, and highest concentration, however, in these cancer cells, you can see that clear vesicular pattern, showing an exaggerating endocytosis response for our cancer cells. In our time course study, we were able to confirm MBV uptake after 1 hour and 30 minutes. We're still waiting to confirm uptake for 15 minutes, and this is an N of 1. We just want to repeat it a couple more times, just confirm the data we're getting. So for conclusions, we've concluded the MBV reduced confluency and cell viability, especially in our neoplastic cells, and that apoptosis is contributing to this process at least somewhat. Esophageal epithelial cells can uptake MBV, and that internalization and uptake can begin as early as 30 minutes. For future directions, we want to further characterize the form of cell death caused by MBV and inhibit mechanisms of cellular uptake to see how exactly the MBV are getting into the cells. Acknowledgements, I want to thank Dr. George Hussey, my PI, as well as Dr. Daniela and Dr. Catalina, and Christine Shen, and everyone else in the Battle at Hussey Lab, as well as the Doris Duke Foundation and the UPMC Human Cancer Center. Any questions?
I have a comment and then a question. So I'll do my comment first. Your uptake photos look amazing. They look beautiful. We do 6 hours and you do not look as good as yours. Oh, good job with that.
Thank you.
And then my second question is, how did you determine the dosages of each? Is that based on a previous study or?
We have a protocol that says to use around 1 like 1 times e to the 11. And we did different concentrations just to dilute it to see if the concentration would affect it. But we did differences in concentration based off what our original protocol says, which is 1 times e to the 11.
Did you do any characterization of the? Well, I guess I do. So, characterization of the.
I honestly don't remember, but I do know after every time we isolate MBVs, we put them through a nanosite so we can see like do these fit the characterizations for MBVs, and if not, we don't use them.
So one, I'm thinking if you showed where like the pill amount and how it is. Were these all from like the same isolation? They were all same or were they three?
These are all from the same batch of labeled MBV. Like I said, I'm not sure why the 50% aren't. It might honestly be issues with like the staining maybe, but I'm not sure why. We're thinking it's something to do with the cell cycle.
I've got a question for you. So why, this could just be a guess, but why do you think cancer cells would be prone to increased uptake of MDB versus the non-cancer cells? Like what kind of advantage cancer cells?
I don't think it's so much an advantage to the cancer cells and so much for like the function of the MBVs themselves. They've shown like when I talked about the therapeutic effects they had, their job is pretty much in control of like homeostasis and tissue remodeling and regulation. So it may not be the cancer cells themselves needing the MBVs. It might be the MBVs that are identifying the cancer cells and are going to them and causing them to be uptaken more.